Welcome, everyone. Thank you for joining us for today's webinar, Comprehensive NTRK Fusion Detection, Agnostic of a Fusion Partner for Optimal Identification of Rare Genomic Events. It is presented by Phillip Febbo, MD, Chief Medical Officer at Illumina, and Maude Champagne, who is in Clinical Oncology Marketing at Illumina. My name is Judy O'Rourke, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Illumina. For more information about our sponsor, visit www.illumina.com. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speakers will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Phillip Febbo and Maude Champagne. I will now turn the presentation over to them.
Thank you, Judy, and welcome everyone for today's presentation. And I'm excited to talk to you about approaches to detecting fusion and specifically novel fusion partners. Increasingly, identifying fusions that can be targeted by drugs is becoming part of oncology and important for oncologists to understand. My part today will be broken down into three specific topics. The first is the role and importance of comprehensive kinase fusion detection. We'll go over some data that supports my previous statement. Two, we'll go over the best methodology for comprehensive kinase fusion detection. What are the methods currently used? What are their benefits? What are their challenges? And finally, we'll go over comprehensive genomic profiling for rare kinase fusion detection. So let's start with the first part, and that is really the importance of identifying kinase fusions in tumors for your patients. It's important to start with what is a fusion. This is very basic, but a fusion is basically when one part of a chromosome exchanges locations with another part of a different chromosome. This can also occur within chromosomes, but the diagram here shows it's simply an exchange, an equal exchange of genomic material between locations. What happens with that exchange is oftentimes two genes that are often in different locations are now placed adjacent to each other. Fortunately, we do see common fusions. About 35% of all fusions are recurrent and known. This diagram demonstrates what happens when those fusions occur. So in this cartoon, you see one gene in blue, one gene in purple, and the fusion, the translocation of the chromosomal segments bring the two genes together so that there is a 5' partner and a 3' partner. Oftentimes, with most of the fusions we're talking about today, the 5' partner is largely acting as a promoter. Largely, these are genes that are oftentimes expressed at high levels in the specific tissue type from which the cancer is derived. And the 3' partner, the kinase domain, is an active element of the protein, oftentimes a kinase that elicits a signaling, oncogenic signaling, that drives tumor development, growth, proliferation, and occasionally metastases. So this novel fusion creates a combination where this kinase is now oftentimes expressed at very high levels, or at least expressed in an inappropriate context, where it has an effect on cancer development and cancer progression. So now that we know that fusions, what fusions are, and how the 5' promoter, when combined with a target kinase, increases its expression, how common is it in metastatic disease? This paper from Memorial Sloan Kettering and their clinical testing that they performed looked at 10,000 patients, and they found that 15% of patients with metastatic disease had fusions within their genomes. These fusions were associated and driving the expression of genes that are known to be involved in tumorigenesis and also can be targets for treatment. When another group looked at the TCGA samples, these are samples from across different types of cancer, been a very large effort over the past decade to molecularly characterize a large number of tumors, of the common and less common tumors. And these investigators asked the question, what proportion of each type of tumor had fusions? This diagram shows you the range with which fusions were identified. Up at the top, you see prostate adenocarcinoma with a high frequency of a specific fusion. And then throughout the other types of cancer, you see different proportions that contain fusions. In fact, when they looked at these fusions in aggregate, 6% of the fusions identified could be targeted with drugs that were available. Again, showing that it's very important to identify these fusions for your patients, that these fusions can be detected across a range of different types of tumors, and that when fusions are identified, a certain proportion are targetable with drugs that are available. Now, each fusion can have different partners. So each kinase that's involved in a fusion can have different partners. And here we're going to talk about ALK fusions, because ALK fusions are probably the first and most widely known fusion where there are ALK inhibitors that have had a major impact in patients with lung cancer who have those fusions, and has dramatically improved the outcomes of those patients when a tumor with an ALK fusion is targeted with an ALK inhibitor. So we talk about ALK fusions focusing on ALK, but the truth is there are multiple 5' partners with ALK that are driving the expression of ALK and driving the dependence of that tumor on ALK. EML4 in bold in the upper panel on the right is the most common and often the example that people have in mind when they think about an ALK fusion. But you can see other fusion partners that have been associated with ALK, and each of those partners drive the expression of ALK and can be targeted with ALK inhibitors with good effect. As we've seen more and more recurring fusions, we've seen similarly that other fusions that can also be targeted have multiple upstream partners. So RET, which is being recognized as a targetable kinase, has been found to have 13 different 5' partners. NTRK, and there are 3 NTRK genes, 1, 2, and 3, is more promiscuous with 61 different upstream partners of NTRK driving the expression of NTRK 1, 2, or 3. And then PGFR alpha, PGFR beta, you can get a sense that even though these kinases are starting to be seen as recurrent specific kinases that can be targeted, the upstream drivers, the upstream partners of the fusions can be quite different and diverse. We'll dive into that a little bit. Again, looking at TCGA data, another group asked specific questions about the frequency of fusions and how common it is to find fusions with the same 2 genes. So some of these numbers are pretty impressive. So 50% of recurrent kinase fusions, so those kinase fusions that were found in 2 or more tumors in the TCGA data, included a new kinase partner, so a new partner for that kinase that had not previously been identified. So you don't always know what you're looking for when you need to identify a fusion. 8% of the recurrent fusions found in a tumor type had not been previously described in that tumor type. So if you were only looking for what you knew, you wouldn't even have looked for that fusion in that tumor type. And 11% of recurrent fusions are novel. They had not been seen before. And this continues to be the case. The more tumors that are comprehensively sequenced and sequenced in a way where you can identify novel fusions, the more fusions we are starting to see and understand. If we bring this back to NTRK, this is a diagram that shows the 3 NTRK genes and the different partners that have been found on the 5' promoter region driving NTRK inhibition. So you can see NTRK1 has been found with the most number of 5' partners, but both NTRK2 and NTRK3 both have multiple. Sometimes they share a partner, such as SQS-TM1 in the middle. You can see that has been found associated and driving the expression of each of the 3 NTRK genes. And the colors of in this diagram represent the different tumor types. So you can also get a sense for that these NTRK fusions have different partners and they can be found across a broad range of tumor types. The importance of identifying these fusions has to do with the remarkable impact that successfully targeting the fusions has on patient outcomes. ALK inhibitors are probably the most widely used in lung cancer to target fusions involving ALK. Here, we just call attention to Alcensa as a drug that can target ALK regardless of its fusion partner and has been shown to improve outcomes. That story is primarily in lung cancer. Remarkably, in 2018, Vitrecvi has been approved for patients with NTRK fusions. This is regardless of the upstream partner for NTRK, and it's regardless of the specific NTRK genes. It could be NTRK1, 2, or 3. And it's also regardless of the tumor type. Vitrecvi approval is the first approval for a drug targeting a fusion that is agnostic of the type of tumor. So it doesn't require it being in lung cancer. It can be in a broad array of cancers. As long as you identify the patient's tumor as having an NTRK fusion, they are eligible for Vitrecvi, and the study suggests that they benefit. And of course, we've already gone over, we know NTRK can have many fusion partners, and 61 have been identified to date. So now that we've gone over fusions, let's talk about how to detect them. So as we think about comprehensive kinase fusion detection, there are multiple methodologies to detect fusion in place already. There's FISH analysis, or fluorescence in situ hybridization, IHC, immunohistochemistry. You can use PCR. You can use next-generation sequencing. And with next-generation sequencing, there are different approaches too. There's the Amplicon-based approaches and hybrid capture-based approaches. And finally, you can either start looking at DNA isolated from tumors, or you can look at RNA isolated from tumors. FISH is perhaps the most widely used method to identify complex rearrangements, and specifically is used for ALK, ROS1, and RET. In a study by Memorial Sloan Kettering. They looked at samples that were tested using FISH, and they also performed Foundation 1 comprehensive genomic testing to assess for the presence of fusions. 26% of patients were found to have fusions by FISH and where therapies were available, but 39%, 12 of 31 patients, were found to have a fusion by next-generation sequencing alone. Within these, FISH break-apart probes had missed 2 ALK fusions, 2 RET fusions, and 1 ROS1 rearrangement, all of which have approved therapies for, and all of which you would not want to miss if you're taking care of patients with those tumors. In 6% of the tumors, an alteration was found for which there was not a targetable, not a drug, and then no genomic alteration was found in 29%. So while FISH is often used and is perhaps most often used, this study demonstrates that you can miss targetable fusions if you rely only on FISH. And this gets even more difficult as you consider some of the more promiscuous fusions. So part of understanding and developing a FISH assay involves knowing the probes. In this case, for NTRK that we spoke about earlier, given the promiscuous nature of NTRK and given that there are 3 specific genes that can be involved, each to get a robust FISH assay, you would need at least 3 different tests and potentially more, which, given the limitations of tissue and given the number of additional fusions you want to check for, just becomes almost untenable as the catalog of fusions increases to keep FISH as your method of analysis and remain comprehensive. So the benefits of using single gene tests like FISH and including IHC, so as we discussed, oftentimes these fusion partners lead to a rearrangement and the overexpression or inappropriate expression of a specific gene. You could take an approach saying, well, if I know what the target of the fusions are and I know what I can drug, I can look at expression. Well, immunohistochemistry requires that you find a strong antibody that can detect the protein and robustly detect the protein. The advantage is it's fast, it's quick turnaround time, and it's low cost. The challenge is that with each additional fusion partner that we identify and each targetable fusion partner that we identify, you need to develop and validate another assay and run it on samples. So it becomes, again, increasingly difficult as our catalog of fusions is growing to stay focused on assays that look at specific genes. RT-PCR is a way that you can start to look for multiple partners using a technology. Again, there's a lot of advantages to PCR with respect to speed, with respect to turnaround time, with respect to cost. Reverse transcription PCR, or RT-PCR, starts with RNA, creates DNA, and then you design probes that can capture either side, either partner of the fusion, and will only amplify a product and only detect an amplified product when the fusion's present. So you can anticipate with this technology, you can come up with multiple probe pairs that capture the fusions that you know. But you do have to know the fusion partners, because in order to get amplification, you need both a 5' and a 3' probe that will anneal to the appropriate target and start the amplification process for detection. Thus, the challenges occur with fusions that have novel partners or unknown fusions that you will miss and not be able to detect. Next generation sequencing is an emerging method that is being applied to fusion detection. As we discussed, there's Amplicon-based next generation sequencing and hybrid capture or enrichment-based next generation sequencing. Amplicon-based is similar to the approach that I just described with PCR, where you take probes for specific regions and you amplify up segments of DNA in order to have sufficient copies to perform robust sequencing and get the sequence of the amplified products. Again, this requires an approach where you understand the regions that you are targeting to amplify. So oftentimes this is most frequently applied to areas where there are specific mutations that are recurrent. Some of the, for example, the activating mutations seen in EGFR, where you can design probes to cover the region where these mutations occur, and you can then sequence those amplified products to see if the tumor has a specific mutation in that area. But as we've discussed with fusions, as you have different partners and as if you don't know what the fusion partners are, it becomes difficult, if not impossible, to design the probes such that you get an amplified product that you can sequence, and that limits the comprehensive detection of fusions. Alternatively, hybrid capture includes creating probes for the gene or genes of interest, regardless of adjacent material, and pulling them down and then sequencing them. So all you have to do is know that you're interested in a specific gene. If you pull that gene down, regardless whether it's in a unaffected and in its native state, or whether it's affected with a translocation and a fusion, you will still pull it down and sequencing. And when you sequence it, you will capture the information adjacent to it, which could include a fusion if it's possible. So the hybrid capture has advantages in that you do have to know the gene of interest, the kinase of interest, and we know all the kinases that are there, and they are included in most of the comprehensive genomic profiling that occurs. You do have to know those genes, but you don't have to be aware of potential fusion partners that are adjacent to that kinase of interest in order to capture that. This figure diagrams some of the differences. So Amplicon, if we take ETV6 and NTRK3, if you know both partners of the fusion and you can design those probes, you can get amplified signal, you'll sequence that, and you'll detect the fusion. With a novel fusion, in this case BTBD1, with NTRK3, you wouldn't have a primer on that 5' partner of NTRK3 and you would not detect it. If you look at the hybrid capture approach, you can design probes that specifically overlap for fusion events and pull them down specifically. But as long as you have probes that target specific kinases like NTRK3, you will still pull down and still be able to sequence and identify that upstream 5' partner that is novel. And that's the advantage of hybrid capture over Amplicon when it comes to specific fusion detections. So hybrid capture does decrease the risk, the need to identify the 5' upstream partner. But when we think about fusions, most of the time we're thinking about the effect of the protein that's created and the RNA that's created, where the upstream promoter is driving the kinase expression. At a DNA level, these regions can still be far apart and interspersed with introns. So these large intronic regions, specifically that have been observed in NTRK 1, 2, and 3, can still challenge the hybridization approach. If you pull down one area of NTRK, the subsequent fusion may be too far away to be captured within a sequencing, the sequencing run. So one approach to limit that challenge is to look at RNA, where RNA, and starting with RNA and the specifically the processed mRNA, brings the two partners closer together by removing the intron material, and you can pull that down more efficiently and capture that intron in a sequencing reaction. So as you think about comprehensive kinase fusion detection, you have to think about what your goals are for testing that patient. And if your goals are to comprehensively assess for fusions, and you want to allow for the detection of novel fusions, and you want to overcome the challenges of large intronic spaces that can disrupt the ability to sequence the fusion and the association between two genes, a 5' partner and the target gene, then you need to, it's pretty clear you walk down to next generation sequencing being a solution, hybrid capture having advantages over Amplicon, and hybrid capture applied to material from tumor RNA having advantages over applying it to material from tumor DNA. Okay, so now we're going to go into the third part of our discussion today, and that's comprehensive genomic profiling for rare kinase fusion detection. As I mentioned earlier, in November 2018, the FDA approved Vitrecvi, or larotrectinib, as the first ever TRK inhibitor for patients with advanced solid tumors harboring the NTRK gene fusions. The important and novel element of this approval is that this is the first time that a drug received its first approval for what we would term a PanCancer indication, meaning it doesn't matter what type of cancer the patient has, as long as they have an NTRK gene fusion with either NTRK 1, 2, or 3, and regardless of the upstream 5' partner, they are eligible for this treatment. And we'll go over this a little bit and some data about what happens when you target patients with NTRK. But from the studies that led to the approval, there was a 75% overall response rate for patients when treated with larotrectinib who had NTRK gene fusions, and that was across adult and pediatric cancers. So the clinical studies for NTRK fusions and the studies that led to the approval found NTRK fusions over 19 different tumor types. It was found in both adult and pediatric cancers, and it was found into up to 1% of solid tumors. So almost any patient that has advanced disease has the potential of harboring an NTRK fusion within their tumor. There are some rare types of diseases, rare types of tumors, where the frequency of NTRK fusions was over 90%. But in others, and some of the more common tumor types, the frequency was about 5%. So understanding the distribution of NTRK and the probability of any patient having NTRK is important to understanding what testing is required. The good news is that oncology is progressively moving more towards comprehensive testing. It certainly has evolved over the past 20 years, where initially we started finding specific single genes and enriching that, and the presence of amplifications or overexpression would be associated with benefit from specific therapies, with HER2 being one of the first that really the benefit of therapy, of the HER2 targeting therapy, was dependent on identifying overexpression or amplification of HER2 in breast cancer. Those different targetable events stacked up. We saw EGFR, we saw BRAF, we saw an increasing number of variants that, when detected, could be targeted and could identify patients who specifically benefited from targeted therapies. And where oncology is moving is to getting to a critical number of those variants where it no longer makes sense to test the variants one at a time, and it makes more sense to do a panel, sometimes targeted panels, but more and more moving to comprehensive panels. Now, what's driving toward those comprehensive panels? And it has a lot to do with events like NTRK fusion partners, like microsatellite instability, or MSI, where increasingly there are variants that have shown to have value and to be targetable regardless of the disease of interest. So that's an important part of this. The comprehensive genomic profiling, here's a definition that comes from CMS, and it is basically defined as a single test using tumor tissue only, not matched tumor normal, that does not distinguish between somatic and germline alterations, and can detect the following classes of alterations. So base pair substitutions, insertions, and deletions. Those have been incredibly important for activating different oncogenes like EGFR, like BRAF, like a large number of others that were increasingly targeted. But as we've talked about, also copy number variation and homozygous deletions, as well as the translocations or the fusions that we've been talking about today. A comprehensive genomic profiling and these rigorous tests can identify each of these types of variants within a tumor, sometimes using the DNA to identify those variants, sometimes using the RNA to identify the variants. Of course, it's important to consider, is comprehensive genomic profiling too much? Are we, using a sledgehammer to hit a finishing nail? And I would argue at this point it's not. The reason is that many of these cancers for which we want to understand the molecular events that are driving that cancer have rather limited tissue. In some cases, as many as 30% of patients with lung cancer do not have sufficient tissue for more than a few tests. And if you go by single tests, you exhaust the tissue quite rapidly. So comprehensive genomic profiling helps minimize the burden on tissue. It can improve turnaround time rather than serially testing one gene at a time, and if it's negative, going on to the next. That prolongs the diagnostic odyssey and can prolong the time that it takes to get a patient on an effective therapy. And all that can translate into a cost savings where you're efficiently testing for the broad genomic events that can be targeted and by targeting can improve outcomes without causing undue delay or incremental cost. While when compared one-to-one, the cost of comprehensive genomic profiling may be more expensive than a single test. If you add up the number of individual tests, it's very quick. Do you get to a cost that's actually more than the comprehensive genomic profiling itself? And this is becoming standard of care in the National Comprehensive Cancer Network. In their guidelines for non-small cell lung cancer, they actually are recommending the comprehensive testing. In this case, broad molecular profiling is the wording that they're using. And they specifically call out that this is important because to minimize the wasting of tissue, because many of these lung cancer patients do have tissue that's inadequate for there's not enough tissue to do a large number of sequential testing. And broad molecular profiling, so you want to spare the tissue, you want to get the broad molecular profiling, but you do want to have the opportunity to identify those fusions like NTRK fusions. And this is a diagram you may have seen before. It is from the study supporting the approval of larotrectinib for PanCancer indication. This is a waterfall plot. It shows patients' best response to therapy with respect to measuring measurable disease. The different colors are different types of tumors, and every patient treated has an NTRK fusion in their tumor. And this is what contributed to the 75% objective response rate and incredible responses across diseases, some of which are very difficult to treat with conventional therapies such as chemotherapy. So this, as well as many other studies, have frequently demonstrated that although rare, when detected and targeted, fusions can have an incredible impact on that patient's outcomes, because so often these fusions are key drivers to the disease, and when targeted, the disease has a remarkable effect and a response. Let's talk a little bit about a single case of a 13-year-old girl. She was diagnosed with a sarcoma, and quite frankly, the sarcoma was, putting her in harm's way. It was wrapped around her aorta, and it was removed through a high-risk surgery, but like many sarcomas, had already spread and next showed up in her spine. Next generation sequencing was performed, and a NTRK2 fusion was detected. And apropos our earlier discussion, it was in fact a novel fusion with the STRN gene. She received larotrectinib, and the tumors had a complete response to that therapy. So again, she's now a 14-year-old freshman in high school, and having had a great response. And more, the future of oncology is finding molecular events in a tumor that are drivers that can be targeted with a drug and having outstanding responses, like what we see with Brianna in this example. So in conclusion, we've talked a lot about fusions, about novel fusions, about how fusions can be found in a broader range of cancers, and that identifying those novel fusions can lead to targeted therapy and outstanding responses. So hopefully today's discussion makes all of us really excited to identify these fusions and not want to miss a single patient, and provide opportunities like larotrectinib has if you're able to identify an NTRK fusion. Hopefully you also now understand that while you have a workflow and using RNA to comprehensively assess for fusions like NTRK, having the same assay look at DNA and identify other variants that can be targeted helps preserve tissue and ensure that you get to a timely diagnosis as soon as possible. Finally, you really taking a comprehensive assay and applying it to your patient with different types of cancer allows you to identify these pan-cancer variants like NTRK fusions, like microsatellite instability, where the patient will benefit regardless of the type of cancer they have. So we've spent a lot of time today going over a comprehensive review of different approaches to cancer and to detect the different types of variants that occur, with the goal that every patient has the opportunity to fully benefit from a growing list of targeted therapies, specifically including those therapies that target kinases that are part of fusions and that can have a dramatic impact on their outcomes. Thank you for your attention today.
We'll look into comprehensive NTRK fusion detection, Agnostic as a fusion partner for optimal identification of rare genomic event using TruSight Oncology 500. The ability to detect novel fusion truly matters. We'll look here into the data from a study from Memorial Sloan Kettering that had looked into their experience with comprehensive genomic profiling on 10,000 patients. What they found is that 35% of all fusions identified are kinase gene, like ALK, NTRK, RET, ROS1, amongst others. Of those, 19% of kinase fusion involved a novel partner gene. So being able to detect all of the fusion partners is truly important. As they use an Amplicon-based chemistry, this 19% of novel fusion would have been entirely missed. It is important in the assay selection to ensure that it can comprehensively detect fusion detection agnostic of the fusion partner. Today we will talk about the latest addition to Illumina's oncology portfolio, the TruSight Oncology 500. It enables detection of DNA and RNA for comprehensive genomic profiling. TruSight Oncology 500 can detect immuno-oncology biomarkers such as microsatellite instability and tumor mutational burden. So it achieves highly confident results. It can save time, money, and sample through consolidation into a single assay. And it addresses the need of oncology today and tomorrow. So let's look into the immuno-oncology biomarkers, starting with tumor mutational burden. A tumor cell with high tumor mutational burden may have a high antigen load, which in turn can lead to increased response to immunotherapy drugs. So not every tumor responds to immunotherapy, but high tumor mutational burden has been linked to better responses. Therefore, the interest in querying every tumor cell or tumor cells for tumor mutational burden load. It is an emerging biomarker in the NCCN guidelines, but it's definitely been of high interest in pathology labs. Now, the gold standard for tumor mutational burden scoring is whole exome sequencing tumor normal. So in this slide here, you see how TruSight Oncology 500 compares to the gold standard using 108 FFPE tissue sample across different tumor types. So what you see here is a concordance between both assay of over 93%. More importantly, it's looking into the accuracy of tumor mutational burden, especially as we look into low tumor mutational burden score. So the clinical threshold that has been suggested today is around 10 mutations per megabase. And the accuracy and determining the appropriate TMB is definitely a little bit more challenging as we're getting into lower scores. So what you find here on the right side of this slide is the performance of TruSight Oncology 500 and the overall agreement with whole exome sequencing tumor normal at a 10 mutation per megabase threshold. And what you can see is that 95.4% overall agreement has been found. So we're definitely quite happy with the performance of TruSight Oncology 500, especially as it compares to other commercial assays as a surrogate to the gold standard, which is whole exome sequencing. Another biomarker that is relevant in immuno-oncology is microsatellite instability detection. Traditional method is to use a PCR assay for MSI detection. What we did here is comparing the accuracy and concordance between PCR assay versus TruSight Oncology 500. And it has shown 98% accuracy using FFPE samples. What you see here is that TruSight Oncology 500 compares is basically looking into 130 homopolymer in the Intronic regions. So a lot more real estate is actually queried versus what a PCR would do. The concordance still being very high with traditional methods, But it leads us to believe that maybe querying additional regions of the genome using next-generation sequencing can be relevant, especially for tumor types outside of colorectal cancer, which has been traditionally queried using MSI-PCR assays. TruSight Oncology 500 allows us to achieve highly confident results. Let's look into a few aspects that makes its performance unique. So it is highly tolerant to sample degradation, which is very relevant when we're starting with FFPE samples. What you see here is how habit capture-based assay can generate high yield even from highly degraded samples. So in delta CQ samples over 5, so highly degraded, we've seen a number of them still having very decent coverage to be able to get meaningful results with TruSight Oncology 500. This is especially relevant as we're comparing Amplicon-based methodology versus hybrid capture. What we know is that Amplicon-based methodology would potentially require all the way up to 4 times larger fragments to be able to have enough space to have primers bind on either side of the region of interest. So as we're looking in oncology to query some FFPE samples that are highly degraded, the ability of hybrid capture methodology and TruSight Oncology 500 more specifically to query degraded sample is very meaningful. We're seeing some of the performance for small variant detection, looking into variant allele frequency of 5% and lower. So what you can see here for different variant types is that the variant allele frequency can be found with great reproducibility amongst even a lower spectrum of VAF across samples. More importantly for the topic of this webinar here is the accuracy for fusion detection. So hybrid capture chemistry, and starting from RNA more specifically, definitely provides a more comprehensive view into fusion detection agnostic of the fusion partner. So how does it work? The probes are basically pulling down the gene of interest, whether it's Intrac 1, 2, or 3, and it's sequencing either through the breakpoints identifying the fusion gene partner, or identifying each gene on either side of the breakpoint and then bioinformatically associate them together. So both known and novel fusion partners can be identified using hybrid capture chemistry, giving us the reassurance that, for instance, across the 61 different fusion partners of Intrac, all of them can be accurately and comprehensively detected. So here's an example just to illustrate a little bit more the point and the difference in chemistry. Using an Amplicon base, it would basically require having a primer to bind on either side of the breakpoint, therefore only being feasible if we know what the partner gene is. So what you see here is an example with Intrac 3, ETD6, of course, being one of the most common fusion partner. It is likely to be accurately detected using an Amplicon-based assay. But some of those fusion partners that may be less common would require having a primer to be able to detect them accurately. For enrichment-based technology or hybrid capture, this is not necessary since the gene is pulled and it will sequence either through the breakpoint and being able to identify that fusion partner. So here's a quick comparison between different assays that you will find on the market today. Again, looking into the plus and minuses of different chemistries. So if you look into the ability to detect Intrac 1, 2, and 3 fusions, as well as the resistance mutation from the DNA portion, we look here into certain Amplicon-based methodology like the Oncomine portfolio, V1, V2, that have Intrac 1 and 3, but missing Intrac 2. You can see that the V3 and the Oncomine focus are actually capturing Intrac 2. But the portfolio is based on Amplicon detection. Therefore, when we're looking into the potential 61 fusion partners that we know of today with Intrac, it would be something very challenging to be able to capture comprehensively. So we know that some of those fusion partners are very likely missed in this case. Archer FusionPlex Intrac panel is using RNA detection and an Amplicon method. They do have a proprietary chemistry enhancement that provides them with an ability to detect novel fusion as well, which is interesting. But in this case here, the resistance mutation would need to be picked up from a DNA assay, which is not part of the solution. TruSight Oncology 500 is the only assay here able to detect comprehensively agnostic of the fusion partner, NTRK 1, 2, and 3, as well as the 4 resistance mutations that are on label with larotrectinib, an NTRK inhibitor. So to bring another example here, looking into a commercially available Amplicon-based assay, out of the 61 fusion partners that we know today, only 24 of them can be accurately detected by this Amplicon-based assay. It means that 60% of NTRK fusions are actually missed. So definitely the advantage of going with a comprehensive NTRK fusion detection tool like TruSight Oncology 500 is very important to be able to capture every NTRK fusion. TruSight Oncology 500 enables consolidation of testing in a single assay, saving time, money, and sample. So it can detect single nucleotide variants, insertion, deletion. In the next few months, it will also be able to detect copy number variation. Of course, it can detect fusion, splice variants, as well as the immuno-oncology biomarker, microsatellite light instability, and tumor mutational burden. And all those targets are within a single assay. The content of TruSight Oncology 500 includes what is found in Foundation 1 and the MSK Impact, as well as TruSight Tumor 170 from Illumina as well. So it does query 523 genes across DNA and RNA. Those genes are relevant for multiple types of cancer. You can see it on the right here across lung, melanoma, colorectal, and much more. It does include the 2 PanCancer biomarkers that are relevant today. So it is NTRK 1, 2, and 3, as well as microsatellite light instability, as well as the immuno-oncology biomarker tumor mutational burden. So all this information is packed into a single assay that makes it widely comprehensive. TruSight Oncology 500 workflow goes from FFPE specimen requiring 5 FFPE slides only across sample preparation with extraction of DNA and RNA into the library preparation that is either manual or can be automated, and then onto sequencing on the NextSeq platform or the NextSeq DX in a research mode, as well as shortly to be available on the NovaSeq as well. The secondary analysis is done using proprietary algorithms from Illumina that confer a better unmatched accuracy for tumor mutational burden scoring, as well as removal of artifacts in SNV, being able to provide a clean and accurate variant calling from our secondary analysis. The tertiary analysis can be done using a third-party platform, such as GHDX, which is a recommended partner for interpretation. TruSight Oncology 500 also addresses the need of oncology today and tomorrow. It includes unique molecular identifiers that are, of course, helping with the accuracy and SNVs, as well as the accuracy and tumor mutational burden scoring, but it is also enabling a development roadmap on liquid biopsy for TruSight Oncology 500. So you can see here some of our early data looking into tumor mutational burden from blood, which is an exciting novel application that is under development at Illumina. So to summarize, TruSight Oncology 500 is a comprehensive fusion detection with this ability to detect fusion agnostic of the fusion partner. But it is a lot more than that. It can detect immuno-oncology biomarkers such as MSI and tumor mutational burden. It provides highly confident results for highly degraded samples, as well as with a low variant allele frequency. It helps with consolidation of testing to be able to optimize tissue preservation, saving time, money, and sample through performing a number of different queries in a single assay. And it is addressing the needs of oncology today as well as tomorrow, especially looking into the future with tumor mutational burden. TruSight Oncology 500 is the backbone of two companion diagnostic agreements that we have, one with Loxo Oncology, Eli Lilly, and the other one with Bristol Myers Squibb, to be able to provide a road towards an IVD companion diagnostic in the future. So on that, I'd like to thank you for your attention and would invite you to visit the Illumina website for more information on TruSight Oncology 500. Thank you.
Thank you, Dr. Febbo and Ms. Champagne, for your informative presentations. I would also like to thank Illumina for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 27, 2019. And as a final reminder, our speakers will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.